I'm doing people right? Yeah. We have like a team. We start up like we'll bring it home for reasons.
It's not just the bias, it's also one
And it is a literal heavy.
One there.
It has like cannoli. It's not.
It's like
I know all the secrets. And you like put it on top of the.
All right, everyone, let's bring it in. Welcome to Thursday. That's the last day of the month. I hope everyone's doing well. Feel like you're getting settled into your fall semester. Of course you are, right? We all have it together all the time. 24 hours a day. Organized chaos. All right, so I'm glad you're still with me. And I hope you're enjoying the topic in the class, despite me. I hinted at this a good bit last time, but where we're going to go today is talking about innate and non-specific immunity. And so these next couple of lectures are going to be very, very post-centered. But before we get started, there are just a couple of logistic details that I just wanted to cover very quickly. One is that I'm going to be posting a homework, the first homework. I've been a little slow in getting that posted, mostly because I've been trying to figure out how to actually post everything in Canvas so you guys can answer the questions in Canvas, as opposed to writing a document and submitting it and having it be a lot more complicated than it needs to be. So I'm going to try and get that knocked out this evening. At least by tomorrow morning, you'll have your first homework assigned. The homework is due on the day of the exam, and so the way that I generally do things with this homework is that some of the exam questions, should you have focused and worked hard on the homework, should not be terribly surprising on an exam. So the big question, are you going to get answers to the homework before the exam? No. So, the types of questions that I'm asking in the homework are similar types of questions that I'd be asking you to answer with the written part of the exam. That said, look out for that being posted, and it will basically cover material up to the last lecture before the exam. Now, what I do in this course is that all of the exams are on Thursday, and the last lecture I'll be testing you on is the preceding Thursday. And what we use that Tuesday for is a discussion of a primary literature paper that is, in my eyes, encapsulating a lot of the things that we've talked about, and that hopefully, even though this is someone's really focused research on a particular topic, that you should be able to feel equipped with enough information to get through this paper and be able to pick up on the big salient points. And we'll have a group discussion about that paper the lecture day preceding the exam. And during that same period, I'll also try and give a quick rundown on things that I think are key concepts from the preceding lectures leading up to that exam. So I hope that seems fair. It's giving you a week of time to ruminate and digest the 6 or so lectures we've been through before the first exam. And so it's posted on the syllabus, but I just also want to reiterate that my office hours are Tuesdays and Thursdays from 3 to 3:45, so basically right before this class. If you can't make those, I know some of you are in a lab that kind of butts up right against this lecture. If that's the case, shoot me an email. I try and be flexible with you guys. This is not a massive class. I will try and make time for you, okay? So don't let your questions go unanswered. It's my big plug. So before we head into this, are there any questions from last time? So did everyone just hear like exam and like just had the like, blood rush from your face? Like, oh God, I totally forgot about this part of taking the course. Don't worry, you will be prepared. Okay, I don't like people being unprepared. So that said, we're gonna get into lecture 4. Now on Canvas, you should also see a kind of laundry list of links to different things relating to this topic. And so this topic in and of itself is a massive topic. It is enormous. It is an enormous research area. A lot of the things that I'm presenting to you today are going to be very high level, and that's on purpose, because getting into the nitty gritty of all of these systems, it's the purview of an entirely different class. And some of you are in that class. You'll learn a lot more about what I'm going to tell you about. But what I want you in this class to have is basically the tools to talk about pathogens and their interaction with the host in an effective way. So these next couple of lectures are going to be sort of the last couple of bits that are going to prepare you to get into the big questions when we start talking about pathogens specifically. So, what are we going to talk about today? Well, we're going to talk about a number of aspects, the key aspects relating to bacterial pathogenesis that have to do with innate immunity. And those are humoral immunity, and as its name implies, these are things that generally have to do with bodily fluids, so blood and lymph. And also, we'll talk about cellular immunity. And so I've given you a couple of glimpses of this in the last lecture, that there is this kind of contrast in what's going on, whether these are sort of very chemical mechanisms or whether they're very cellular mechanisms. And then essentially, how are these cells that are involved in innate immunity, how are they recognizing things? How are they understanding what's good and what's bad? I'm going to give you a very cursory, big overview of how they do that. And then what are they doing to elicit responses from our bodies to combat pathogens? Okay. So we talked about this last time, that there are these categories of innate immunity, that there are anatomical barriers. So we spent a lot of time talking about epithelial tissue, your normal flora, where we ended the last lecture, and that these are things that our body, in terms of a multicellular organism, we've basically developed these things to protect our organs and all sorts of other things in us. There are physiological and chemical barriers, so maintenance of specific temperatures and pHs at different places in the body, production of exoenzymes like lysozyme, lactoferrin, and one big physiological chemical barrier we're going to spend a lot of time talking about today is complement. And that'll hopefully be clear what that is in a second. Phagocytotic and endocytotic barriers, so basically being able, we have cells in our body that are surveilling around and looking for things that should not be there, looking for pathogens. And inflammatory barriers, so recruitment of these phagocytic cells, so aggressive recruitment of these particular cells to specific locations in the body, and we'll see what some of those cells are. So, humoral immunity. What is this? So, humoral immunity are components of the immune system found in extracellular fluids. So, things that are found in blood, lymphatic fluids. So, what's in this? what are these components that are important for innate immunity? This is a much longer list, but the ones that are important for us, antibodies, and we'll see where those come from next time. But they're important today in our discussion of innate immunity because they're a component of innate immunity, even though they are really in the realm of adaptive immunity. And so it's one of these interesting molecules that sort of bridges two worlds that we like to divide things into in terms of immunity. We talk about our immune system, that there's this innate system and then there's this adaptive system, and that they're really functionally different, and that what we'll see is that there's a lot of overlap in terms of how these things are talking to each other. They're not just completely autonomous systems doing different things. Production of antimicrobial peptides, we'll get into this as we start talking about this host-pathogen interface. So there are antimicrobial peptides present in blood and lymphatic fluid and other extracellular fluids. Clotting factors, so these are really exclusively found in blood. I'll talk very briefly about these. And complement proteins, where we'll spend a good bit of time today. Because complement is one of these things that is an important, arguably one of the most important parts of innate immunity in mammals. And this contrasts with cellular immunity, as these are macromolecules, they're not cells. So these things, of course, are produced by cells in our body, but they in of themselves are not living things. They're chemical, like macromolecules of proteins, and so on. So, not cells. And that's really what is differentiating humoral immunity and cellular immunity. So our first stop today are antibodies. And so, how many of you guys have heard of antibodies? I hope everyone in the room can raise their hand, whether it be from a course here or pop culture, wherever it may be. You've probably heard of antibodies, and you've probably heard that they bind to things very specifically. And both of those things, that you've heard of them, they bind to things specifically, and that they're produced by us and they're important. And all those things are true. But at brass tacks, they're produced by immune system cells. We'll see which ones in a minute. They are present in extracellular fluids, so they're present in your blood, they're present in your lymphatic system, they're present in mucosal tissues. We'll see some of that a little bit later. So they get basically produced and pumped out all over different parts of our bodies that are potentially interacting with pathogens. And they bind very specifically to what we call epitopes. And epitopes are specific chemical structures. And what does that mean? It means that antibodies they're basically, and we'll see why they are specific and whatnot in coming lectures. But basically the idea here is that they are a protein that is produced and secreted by immune system cells, that these proteins have basically a conserved core region here, and we'll see how that's variable across different families of antibodies, but that they have a variable region on these protein structures that is able to bind to specific compounds. And so basically the same pool of B cells that we'll talk about in just a few minutes, each of those different B cells is producing essentially a different version of the same protein in this region. And this region will bind to specific epitopes. And these epitopes can be the charged surface of a protein, they can be specific amino acids, they can be metals, they can be plastics, like things that are non-biological. They are, through diversity in what this variable region of antibodies looks like and what its own amino acid composition is, basically dictates what it's able to see and bind to. And that this can range from biological things to inorganic things to non-biological plastics. You name it, you can generate. You can typically find antibodies that bind specifically to most anything. And that's something that we exploit now in biotechnology, for example. But in the context of the human immune system, mammalian immune system, this is one of the ways that we develop essentially specific targeting of specific pathogens that we encounter throughout our lives. Again, it's important for pathogen. So are there any questions about antibodies? So hopefully some of what I said was kind of resonated with kind of things that you feel like you've heard before or have directly heard before. One thing that you may not have seen before are antimicrobial peptides. And what these are, they are small proteins that are secreted by various cells in our body. And they are essentially, as their name implies, they're meant to somewhat indiscriminately target specific types of cells and bind to them and basically kill them. So, what are they? They're typically cationic. So they're typically small proteins that have lipophilic and cationic, so positively charged amino acids that make up their protein structure. And so what does this do? What's the benefit of these sorts of this sort of charge arrangement that you basically have very neutral kind of lipophilic amino acids, and then you have a whole bunch of positively charged amino acids, things like lysine and arginine. On sort of one face, what does this do? Well, importantly, our cytoplasmic membranes, at least the leaflet that is facing the outside world, is generally not, the overall charge of that surface is not terribly negative. And it's fairly neutral. And one of the benefits of this is that bacterial cell surfaces are, by and large, predominantly negatively charged. So, what would you expect with something that's positively charged and something that's negatively charged if they're close to each other? They'll attract each other, right? They'll bind each other. And so what does this do for cationic antimicrobial peptides? This facilitates strong binding to bacterial cell surfaces. And what you might imagine, once this thing is bound to the bacterial cytoplasmic membrane or outer membrane, if you're a gram-negative bacteria, is that this cationic peptide is going to coordinate with the bacterial membrane, and you get enough of these cationic peptides binding, they'll begin to coordinate with each other and with basically this hydrophilic core of lipid bilayers with these lipophilic amino acids, and essentially pop this membrane. So you'll form these cationic antimicrobial peptides typically either destabilize this membrane or actively form pores in this membrane. And so ultimately what this does is, if you destroy a membrane, what do you think is going to happen to that cell?
Degraded.
They're going to lyse or they're going to die. Things are not good for you. This is going on. So, that said, it's one of the important things that our bodies do to basically combat pathogens and also control the overall abundance of bacteria in specific environments in our bodies. So what's interesting is that oftentimes, what it seems anyway, there's some evidence that we are sensing and responding to what microorganisms are, say, in our intestines, and we're producing corresponding sort of catalogs of antimicrobial peptides to sort of manage different populations of different microorganisms. So cationic antimicrobial peptides is one example antimicrobial peptides, and it's the one that we understand best how they work. There's others that do other crazy things as well. So, who's had a cut? I have. I've gotten lots of them throughout my life. And if you don't put well, even if you put a Band-Aid on, one of the things that you'll notice is that blood clots. And that's an important property of blood. And that is a way that we protect ourselves from systemic infection. And what this process is called is coagulation, and proceeds through a process called the clotting cascade. And this is essentially a transition from the blood being a liquid to a gel. And in terms of gels, it's a very mechanically strong gel, so it's very almost glass-like gel. And so again, this is to keep pathogens from gaining access to deeper tissues that are not, say, keratinized. So if I cut my finger open, I start bleeding, and that clots. Basically now pathogens don't have access to the connective tissue underneath my epidermis and dermis. And so there are three ways that this typically gets activated: through extrinsic, intrinsic, and this common pathway. I'm not expecting you, because I'm not going to go into the details of this. But what goes on with this entire clotting cascade is that it is through a series of proteolytic cleavage events. So you're activating through three different channels a number of proteases. And so this intrinsic mechanism is basically like, if you bang, if I'm remembering this right, if you basically have internal damage and things, surface contact, you basically induce cleavage of factor 12 to factor 12A, which cleaves factor 11 to factor 11A, cleaves factor 9 to factor 9A, which 9A then converts factor X to factor XA, which now and all of these are proteases, okay? So you're basically turning one protease from an inactive form to its now active form, and cleaving the next protease, which activates the next protease. And why do this? One of the important reasons for having cascades of any kind is either because you want to amplify a signal in a biological system. So why have phosphorelays, for example, is that you can amplify signals with relays. You can also do things like exhibit different levels of control. So you might have an activating event, but for the next step to proceed, you might need event A plus event B to move on to the next step in a particular cascade. So it allows you to have more exquisite, fine-tuned control over when events happen biochemically. There's a lot of reasons for it, but just the thing to keep in mind with clotting is that it's a cascade of activating different proteases, and that the main important player in all of this coagulation is factor 10, factor X. And so anything that is essentially transitioning factor X to become factor XA. So factor XA is the active form of factor X. Factor X cleaves prothrombin to produce thrombin, which then induces fibrinogen polymerization to fibrin, and essentially forms a stable clot. And so this is the logic behind all of these different cascades, is transitioning factor X to form factor XA to turn prothrombin into thrombin, which then precipitates production of fibrin clots. So that said, this is not going to be the only proteolytic sort of cascade that we're going to see that's important. So this is important in innate immunity, is it's basically like, I don't know, the example that I kind of use in my head is like, all right, you've got a drafty window, right? And it's not really like the glass window, but it's kind of the joint around the window, and I can go buy this like spray foam stuff from Home Depot or Lowe's and squirt this stuff into a crack. It'll fill up and basically stop air from coming in this gap in my window frame. The same thing is basically going on with coagulation. It's a fluid. It's going to fill this wound. It's going to be exposed to either surface contact, extrinsic tissue damage factors, or basically be exposed to other proteases that are around and activate factor X, which is going to basically cause this liquid blood to turn into a hard gel, and basically block up that. Okay, so any questions so far? Because now we're gonna head into the, I think, one of the trickier topics. Yep.
So, extrinsic, intrinsic, and the common, are those like the different steps of coagulation, or does it happen in different?
There are different ways that you can activate transition of factor X becoming an active factor VIII protease, factor 10 protease. It's like I'm not going to get into like what exactly each of these pathways do because it's not explicitly important for this course.
So is it like the different ways you hurt yourself, like whether it's like a small cut or like a deep cut?
Well,
or
it's
something
basically
that?
like whether or not you are like. It's been a long time since I've actually specifically thought about this, but it's basically like what is inducing this event, whether it be like actual tissue damage, whether it's like blood contacting different like the air. I mean, there's a lot of different things that will trigger a number of different proteolytic cascades that all center around turning factor X into factor XA. And that's kind of the most important thing that I want you to take away. So there's a lot of different ways that this event can be triggered. But there's sort of three major ways that we know how this happens, and that like as we start talking about certain pathogens, they explicitly will do this. If you have a blood-borne infection, there are pathogens that will actively cleave factor X to factor XA to induce clotting. So I just want you to see this bit. Yeah, good question. Okay. So, complement. How many of you guys have heard of complement before? A couple people? Is that because you took immunology? Okay, all right. I'm probably going to butcher this, so be kind. All right. So, complement is a series of proteins that are produced by the liver in mammals, and they are found primarily in blood plasma. And so this is a component of your circulatory system that's delivered to other tissues. And so like clotting, complement activation is a series of proteins that, when activated, elicit an immune response. And this, like clotting, occurs through a series of proteolytic cleavage events. And like most components of innate immunity that are proteinaceous, that are just kind of free-flowing around their humoral, proteolytic cleavage is a big activating mechanism. And so what happens when complement is activated? This results in recruiting phagocytes that we'll talk about shortly, basically immune cells that will just eat up pathogens or other things that are the complement system, for example, has dictated to say, like, all right, this is not supposed to be here. We need to get rid of this stuff right here. It will also directly kill pathogens by disrupting cellular membranes, and that there are three major ways for activating complement, and we'll talk briefly about what those are. So we'll get into some of the nitty-gritty But there are three ways that essentially complement gets activated. Borrowing this figure from a textbook that I think I give links to the sections of this immunobiology textbook that's freely available from NIH, the National Institutes of Health. And basically there's the classical pathway that we'll talk about, the MB-lectin binding pathway that activates complement, and an alternative pathway that really has to do with basically sensing certain aspects of the pathogen's surface, kind of like cationic antimicrobial peptides, except a lot more, a fair bit more specific. And so what happens with complement, and right now I'm saying complement, and for just about everyone, it's this vague, mysterious term that, okay, it's produced by the liver, it's in the blood, it's stuff, and we'll get to exactly what this stuff is in just a minute. But what does it do when complement is activated? It does a number of things. So it recruits inflammatory cells, and many of these cells that are involved in inflammation will be directly involved in a process called opsonization. And essentially what this is opsonization is basically phagocytosis, or basically like performing endocytosis and gobbling up a pathogen, putting them in a vacuole. If you remember from last time, these inflammatory cells that we're going to talk about, they have lysosomes in them, and these lysosomes fuse with these vacuoles that were formed, and we basically have an endolysosome that digests whatever that phagocytic cell has taken up. And so we'll talk about that, and then also just directly killing that pathogen if possible. And the thing that's crazy about complement is it does all of these things, and I'm going to give you a really superficial look at complement, but it is really, really complicated. I believe one of the links that I give is a very nice tutorial video taking you through a number of these different activation pathways. I will not ever ask you specific proteins that I don't point out and say, if I ask you what C3A and C3B that we'll talk about, what they each do, that's something that I want you to remember. You know, what does like C4B do? I'm probably not gonna go after you say it's an question, right? And so, what is the most important process in complement activation is formation of an enzyme called C3 convertase. I'll get to what that means in just a second. But what does C3 convertase do? Well, it cleaves a protein called C3, which is complement protein 3, into two different things, C3A and C3B. And so C3A, what it does is a peptide mediator of inflammation. And so it is an important cytokine that we'll kind of talk about cytokines towards the end of the lecture. But basically, these are protein signals, essentially like protein, almost we could almost call them hormones, essentially elicit an inflammatory response and recruit phagocytic cells to a site of infection. And C3b is a separate protein. Well, it's not a separate protein since it's C3 that's been cut in half. And C3b binds to complement receptors on phagocytes and is critically important for mediating opsonization of pathogens by phagocytic cells. And also, C3b recruits a whole slew of other complement proteins to form what is known as the membrane attack complex, and that this is basically a big pore that forms on a pathogen's cytoplasmic membrane and basically pops this pathogen cell, whether this be a bacterial or eukaryotic pathogen. So, Formation of C3 convertase occurs through 3 separate pathways, and a number of different proteins can come together to form this C3 convertase enzyme. So it's not necessary. So when we're talking about a C3 convertase, what we're talking about is an enzyme capable of performing that proteolytic chemistry of converting C3 into C3A and C3B. Okay? So the thing that's really crazy about these different pathways is that many of the proteins, say the classical and MB-lectin binding pathways, C4 and C2, are common to these pathways, but they're not important for making a C3 convertase by this alternative pathway, for example. So different proteins can come together to make, different complement proteins can come together to make a C3, an active C3 convertase. And this is why we, basically how we're able to functionally pull out these 3 different pathways. So the classical pathway that I'm going to talk to you about is how the complement system basically uses the specificity of antibodies to recognize something that should not be in your blood or in your body. The MB-lectin binding pathway, this is a more general system in that it is actually looking for specific carbohydrate patterns on the surface of different types of cells that are not self. So it's looking for nanos, for example, on pathogen surfaces. And essentially, this is through a family of protein called lectins. So lectins are good proteins produced by us. They're really good at binding to specific or different types of poly or monosaccharides. And when these lectins bind to things, our complement proteins, if these lectin-bound things are, say, in your blood, this is something that your complement system will say, this shouldn't be here, and we're going to form an active C3 convertase and activate this complement system and recruit cell-mediated immune players, or we're going to form this membrane attack complex on the surface of the pathogen. And then this alternative pathway is one of the ways that we sense and respond to different aspects of pathogen cell surfaces in a fairly non-specific way. All right, so in the slides yourself, you should be able to kind of zoom in on these pictures, and this is also an image that is available in the immunobiology textbook link. But this classical pathway, it's a pathway that I want you to see and kind of work through. In that it's important because it's one of the most important ways that, in addition to antibodies binding to pathogens and just kind of inactivating certain aspects of pathogen biology that we're going to talk about, antibodies binding to a pathogen don't necessarily out and out kill that pathogen, right? So it's basically like saying, all right, I'm gonna hitch a great big cinder block to your leg, and maybe you're probably not going to be able to run around and cause problems as well as you would without it. But now, if there's something that looks and says, "Hey, you have a cinder block tied to your leg, and I'm going to eat you," that's a different story, right? So that's essentially what's going on with this complement system, especially when things have antibodies bound to them. So what's going on with this classical pathway is that let's suppose that an antibody is bound to a pathogen, and there is a complement protein 1, that recognizes this, that recognizes antibodies bound onto pathogen surfaces. So it's now, we form this antibody C1 complex. This C1 complex then cleaves a protein C4 into 2 proteins, C4A and C4B, and also a another complement protein 2, that it cleaves C2 into C2A and C2B. And basically what happens is that C4B and 2B come together. And when these two cleaved subunits come together, what do they form? They form a C3 convertase. So the C3 convertase that is now formed on the surface of this pathogen is actively cleaving C3 proteins that are out and about in this whole massive mixture of complement proteins to form C3b and C3a. And so now there's a whole bunch of C3b that's being produced and covering the surface of this pathogen, a whole bunch of C3a that's also being produced and going off and recruiting phagocytic cells and other immune players to show up to this site of infection. And essentially, this is when, for a pathogen, all hell's going to break loose, right? So now, this is a chemical signal, a strong chemical signal, that there's a pathogen here and we need to take care of it. So, the common features of all of these pathways is formation of this C3 convertase. And so there's a video that I've posted that will take you through in much more gruesome detail the molecular events leading up to formation of C3 convertases via all of these pathways. I'm not going to ask you detailed questions about all of these pathways. Again, I think these sort of big waypoints in complement activation are important to understand, and that basically there are 3 different ways to form a C3 convertase. You start converting C3, complement protein 3, into C3A and C3B. C3A does what?
C3, which one?
C3A.
Recruits phagocytes.
Right, so that is the immune response, basically cytokine. It gets produced by the complement cascade. And then C3b, this is one of the things that stays stuck to the pathogen and becomes a receptor for opsonization. And we'll talk just now about what opsonization is. Not the biggest fan of this particular figure, but essentially what goes on and this is showing this MB leptin pathway, but the big point here is that these pathogens are decorated with CD1, and on the surface of phagocytes is a protein, and one of the things that defines phagocytic cell types when we basically looked at what surface proteins are present and what do they do, that there is a protein called complement receptor 1, and complement receptor 1 binds to C3b, and these C3b proteins are stuck to the surface of this pathogen. So now this thing is bound to this phagocytic cell. And remember we talked about there are some other proteins that get cleaved by C3 convertase as well, and one of them is C5, and so C5a is a protein that when C3b has been bound to the CR1 receptor on macrophage and other phagocytic cells, this C5a is now a secondary signal to say, yep, like this is a full-on complement activation event, not just some random thing that has some C3b protein stuck to it. Like time to engage in phagocytosis, take this thing up and destroy it. And that is essentially one of the outcomes of opsonization of this complement cascade, in addition to formation of this very complex membrane attack complex. That C5b is one of the players in forming this, and another C5a is one of the signals for opsonization. So essentially, every bad thing that could happen for this pathogen because of activating this cascade is going to happen. So you're either going to die because of membrane being disrupted, or you're going to get eaten by a phagocytic cell type. When we're healthy, and a pathogen that is not very good at dealing with itself. Which thankfully is the vast majority of pathogens and bacteria that we know. So, this all begs the question, how the heck do we not activate this by ourselves all the time? This is a great question. And it's because we produce a protein called factor H, which is a strong inhibitor of C3 convertase. And so C3 convertases don't form on our own cells. We're healthy, and we're producing factor H. So factor H deficiencies, I don't actually know if they're lethal, but you are likely not going to be a happy individual with non-functioning factor H. And so one of the things that helps our own cells be covered with factor H is that factor H binds to a compound called sialic acid. Sialic acid, yes?
Actually, people who have deficiency of factor H would be cleaving C3 way too often. So as a result, you're never forming C3b, and it's never binding to the pathogen. So they would have problems killing the bacteria. We just did an immunology. What was it? A case study, and the person, his diagnosis was deficiency of factor H.
Okay.
Yeah. So the blood test showed that he was cleaving C3 way too often.
Okay. Right, so he's cleaving this into like C3a and C3b.
Right.
And so you essentially just have like constant inflammation.
Mm-hmm.
Okay, yeah. Okay, so you're not dead. But
No, you're not dead.
You're certainly not happy. Right? So our cells are covered with sialic acid, and this is something that binds to factor H, and is one of the reasons that we don't basically attack ourselves with the system all the time. And one thing that's cute that we'll talk about a little later is that if you're a pathogen, there's this protein that can shut this whole thing down, and you figure out a way to bind it, that's pretty useful. Right? So there are actually pathogens that exploit this and cover themselves with factor H to essentially kind of go incognito and bypass activating any of these pathways directly by just simply blocking C3 convertase from ever forming on their surface or being active. All right. So this leads to our first question of the day, which is antibodies and complement are both key parts of humoral immunity. And what ways do these systems overlap and function? Go ahead and talk amongst yourselves for a minute. All right. So to stay on time today, go ahead and reveal the responses. So both can recognize intracellular components of pathogens. Both are extremely specific in the components that they recognize. Both can recognize extracellular components of pathogens. Both require activation by protease enzymes. Or last, both are components of just the innate immune system.
Okay, so last week. Oh, right, so I'm gonna read this and not make this. Okay, so okay, yeah, definitely, but how are you now? This is a C3B.
Oh, I have it like actually. Don't. Yeah. Okay, I'm gonna go ahead and open the response window. Keep it open for a minute. And it is open now.
What was the first one?
Congrats.
No, I keep.
All right, so let's try and go ahead and get our final responses in. A few more seconds. There are 37 of us today. All right. All right, so predominantly C and D. All right. So, as we've kind of recognized in contrasting these systems, that antibodies, they're really specific, and the complement system is not necessarily that specific. So if looking for general things, in this classical pathway actually kind of piggybacks on the specificity of antibodies. Neither of these, well, both are, one is capable of sensing intracellular components of pathogens. That's not the complement system. And only one of these requires activation by protease enzymes. That's the complement system. And as we've seen, this is like antibodies are one of these kind of interesting things that are going to bridge this gap between the innate and adaptive immune systems. And one of these crossovers is that antibodies and complement work together. Okay? And the answer is indeed C, that both can recognize extracellular components of pathogens. So the complement system is looking for things that are outside of that pathogen's cell. So it doesn't see cytoplasmic stuff, right? Okay. So, are there any questions so far? All right, so cellular immunity. So this is the stuff that is not necessarily macromolecules. And where we need to kind of start with thinking about cellular immunity is the organs of your immune system. And that we, throughout our bodies, in addition to the vascular system, we have kind of the alternative fluid highway, and that is our lymphatic system. And so this is comprised of lymph nodes and your spleen, and all of these are major reservoirs of immune cells. And they're also, and this is at the sort of systemic level, they're also mucosal immune organs, and that these are where the immune cells are localized to mucosal membranes. for example, and that there are also lymphoid tissue that is essentially integrally associated with mucosal tissues. And so these mucosa-associated lymphoid tissues, these are essentially lymph nodes that are like that are part of your intestinal system, for example. And we call these MALT for short. And we'll talk about this a lot. But importantly, these lymph and vascular systems, they're trading fluids a lot. But they are distinct separate systems. And this lymphatic system, this is the highway for all of these different immune systems, immune cells in our bodies. And these are all derived, all of these kind of major types of immune cells that we're going to talk about in this class. And I'm just giving you kind of the big players we're going to talk about. This can be further differentiated into a lot of other things. And I'll introduce these players as we need them throughout the course. But they're all derived from bone marrow stem cells and essentially white blood cells. They all come from the same stock, so to speak. And that there are those that are associated with innate immunity, and these are differentiated cells from myeloid precursors that form dendritic cells, macrophages, neutrophils, and mast cells, and I'll talk about what some of these do in just a second. And then there's the other branch that are the adaptive immune cells from lymphoid precursors that are B cell and plasma cells and T cells. And that these have distinct roles in our adaptive immune system that we'll talk about ideally next time. So, functions of these innate immune cells, dendritic cells and macrophages, what these are doing is that they're the surveillance cells. So they are moving around all the time, looking for bad stuff, and are actively recruited when bad stuff is found. And what these do is that they opsonize foreign cells and material, and that they're intimately involved in the process of antigen generation and basically presenting epitopes to our adaptive immune system. So this is another bridge between this innate immune system and our adaptive immune system. And neutrophils and mast cells, so these are things that are really responsive to cytokines and chemokines. So C3A, for example, is a great chemical signal for these cells. And they're also capable of taking up and destroying bacteria in the case of neutrophils. And they basically, as a result, produce even more cytokines and recruit even more immune cells. Whereas mast cells, they sense these cytokines and chemokines produced by different events in our immune system. And they release a compound called histamine, among other things. And histamine dilates blood vessels, allowing for more innate immune cells to enter infected tissues. And so, in and amongst its many other basically inflammatory functions. So taking antihistamines, for example, blocks the action of histamines. So, it leads to a second question. Are there any questions about these different types of cells? Yes?
So I know macrophages come from monocytes. Do you know where that switch happens? Like, where does a monocyte turn into a macrophage?
Oh, offhand, no, I don't. But what's that?
In the tissue.
Is it in
In the tissue.
the tissue that the monocyte goes to?
Yeah.
Is that right? Yeah. Because a lot of times, like in sort of infectious disease microbiology, we just talk about like monocytes or macrophages, and they're almost like synonymously used. But thanks for the clarification. All right. So let's go ahead and the sequence. So, the question here is there are immune cells involved in both innate and adaptive immunity. However, those lines get pretty blurry. So, in amongst those two different families of cells, of these adaptive immune cells, which one is also involved in aspects of innate immunity? So the kind of the discussion defined a little bit. Are they macrophages? Are they neutrophils? Are they mast cells? Are they T cells? Or are they B cells?
I got it right.
All right, I will open up the response window here. Feel free to keep discussing or let this go until I see around 37 people or so, and I'll pipe up.
To be real quick.
Okay, and I don't see.
All right, I think all responses are in. Let's see what we're thinking. Okay, all right, good. Distribution of answers here. Macrophages are the kind of, or T or B cells seem to be the big players here. So the trick in this question is that I'm asking which type of adaptive immune cell is also involved in aspects of innate immunity. Now the tricky bit here is that the macrophages, these are explicitly, we kind of categorize them as innate immune cells. But they are an important bridge to the end of antigen presentation. Neutrophils and mast cells, these are primarily focused with aspects of innate immunity, and our T and B cells are these immune cells that we categorically associate with adaptive immunity. But B cells are ultimately producers of antibodies, right? And we'll get to that next time. So B cells really end up being this interesting bridge that are where we say something is this is adaptive immunity. It's really also a player in innate immunity. So that said,
So it's E, right?
yes, it is. good thing I'm not grading things this week. So this week, I'm looking to get everyone acclimated to these types of questions. So what I'm really looking at this week is that you're here and you're participating. Okay. So the last thing that I want to get at today is actually a really complicated topic, and it'll come up again. And is basically how are these innate immune cells figuring out what they're supposed to do? So it's one thing to know that you can do stuff, but how do you know when you're supposed to do it, right? And that is where some important players come in the picture. And so how do innate immune cells recognize pathogens? So one of the ways is something we've already talked about, and that's complement activation, because this is something that recruits these cells and promotes opsonization, and essentially you're going to take up and destroy pathogens that you have been molecularly instructed to do. And another is through pathogen-associated molecular pattern receptors. And so what this is that epithelial and innate immune cells possess receptors for common bacterial structural components. And we'll talk about what some of these components are in just a second. But essentially, what happens is that these receptors that are sitting on the cell surface recognize a pathogen-associated molecular pattern. When they bind to these patterns, it initiates production of cytokines and chemokines. And so essentially, when our cells in our body sense things that are programmed, essentially hardwired to look for all the time, those cells start ringing the alarm via production of cytokines and chemokines. And so, what are these PAMP recognition receptors, or toll-like receptors? So you'll see these referred to either as like PRRs or TLRs for short. And shown here is TLR2. And what's generally happening with all of these systems in our bodies with toll-like receptors or pattern recognition receptors. So TLR2 is a pattern recognition receptor that looks for peptidoglycan. So when it sees peptidoglycan, it induces a signal transduction cascade that ultimately results in transcription, translation, and production of cytokines or enzymes that are responsible for producing specific chemical chemokines. And also antimicrobial peptides in cells that are basically wired to produce antimicrobial peptides. So this is one of the things I was mentioning, that we're sensing and looking for essentially what types of bacteria are out there broadly, and how many of them are there. One of the ways that we and produce corresponding responses to that, to either encourage or restrict microbial growth. One of the important ways that we do that is through these pattern recognition, PAMP recognition receptors. And they're fairly diverse. Different animals have different numbers of them, and the ones that I'm showing you here are some of the really important ones in humans. So there's TLR6, TLR2, TLR1, and that these respond to different, predominantly to peptidoglycan, gram-positive bacteria. They're capable of recognizing a number of other substrates as well, so they're not super specific. They're specific enough to look for specific things like gram-positive peptidoglycan, but they'll also recognize other things. TLR4 is one that we'll see a lot in this course, and that it is responding to LPS, which is lipopolysaccharide produced by gram-negative bacteria, and I showed you that in the second lecture, that big lipopolysaccharide that had lipid A and a core O antigen and that long chain O antigen. Lipid A is a strong activator of TLR4. And so if I were to basically take the purified LPS, inject that into you, you would basically have it would kill you ultimately, and then it would kill you through overactivating this toll-like receptor response system. You would start overproducing various chemokines and cytokines, and basically have the worst fever you've ever had in your life, and ultimately die basically of septic shock. And a number of other TLRs are responding to different things as well. So TLR5 is another one that we'll see come up. And that this is actually sensing flagellin. So flagellin is the filamentous protein in a bacterial flagella used for motility in a lot of different bacteria. And interestingly, flagellin is highly structurally conserved. And as a result, it's something that we have something programmed to sense and respond to. So one of the things I think that this is strong evidence for is that we have had a long relationship with bacterial commensals and pathogens, right? And we have a hardwired set of systems to sense and respond to their presence. And we'll see some interesting ways that bacteria work to either overactivate these systems as a pathogen or to avoid being detected by this hardwired molecular innate immune mechanism of pathogens. So, these cytokines and chemokines that I've referred to, they're small proteins produced and secreted by epithelial cells and other innate immune cells in response to sensing things by these PAMP recognition receptors through complement activation, and also tissue damage in general is something that basically surrounding cells, if other cells are dying, these same signals that were causing apoptosis in one cell, the neighboring cell is going to say, wow, I'm awash in these signals that say I should kill myself. I'm going to produce a whole bunch of chemical signals to say that something bad is happening. And so these cytokines and chemokines, these are sensed by neutrophils, mast cells, and other leukocytes, which are kind of broadly those immune cells that we talked about earlier. So we'll be talking about leukocytes. So the idea here is that there's a cytokine producing cell. It senses something in the environment that shouldn't be there, starts producing cytokines. There are cells that have receptors for these cytokines, and when they sense these things, it activates them to perform their programmed biological functions. So what do cytokines do? They recruit leukocytes to sites of infection. Leukocytes such as neutrophils, they begin to attack on cells, often good or bad in the area of infection. So we'll see the activity of things like natural killer cells and things later on. So localized inflammation, this can be good. So that nasty infected cut or whatever you have going on, that thing getting red and painful, that can be a really good thing. So left unchecked, unfortunately, inflammation can become systemic, and this is very, very bad. Okay? And so what we generally term the systemic level of inflammation in the context of bacterial pathogenesis, this is septic shock. And so you end up with septicemia or like blood infection, that becomes systemic infection. This is ultimately what's going to kill. It's not really the activity of this pathogen anymore. It's your body's response to the pathogen is going to become uncontrolled. And so, you sense and respond to this system-wide. All of these cells with PRRs throughout your body are going to sense and respond to this through producing cytokines. You're going to have a massive inflammatory response, and the response is systemic, causing essentially large-scale organ failure, all sorts of nasty things. And so we'll talk about this again when we talk about endotoxins much later in the course. But basically, this systemic level of response is something you never, ever want to have happen. And this is really when, say, like Staph aureus becomes a blood systemic infection. This is what kills you, not necessarily Staph aureus. We actually are our own source of that. So I'm not going to belabor honing through summary bullet points here. So I want to leave just a minute for questions while you guys pack up. I look forward to chatting with you guys next week. Are there any questions?
Yeah, so